Dear BiOdysseans, let's go back over the second part of the bricks of life. The first part was focused on carbohydrates' importance for living beings. Now, we will highlight why lipids, and fat in general, are crucial for life. Lipids are fatty bodies and form a very wide group of biological molecules whose principal characteristic is to be hydrophobic, that is to say, insoluble in water, but dissolve easily in organic solvents such as methanol, acetone, chloroform, or benzene. Thus, if you put oil and water together in a container, whatever their proportions, they will never mix. If you shake enough hardly, you will eventually obtain a shady mixture, but after a while they will separate and the oil will float above the water because it has a lower density. We call water-soluble product hydrophilic and oil-soluble product lipophilic. Lipids are divided in two categories, according to whether they are hydrolyzable or not. Hydrolyzable means that they can eventually decompose to smaller molecules. Let's take the example of the hydrolysis of an ester by a hydroxyl anion that you can find notably in water. This reaction will produce a carboxylate anion and an alcohol. Well, this reaction, also called saponification, is a way to produce soap. Indeed, yes, these two products, the carboxylate anion and the alcohol, are soap. Then of course you can have different kind of soap. For instance, if the hydroxyl comes from a potassium hydroxide, you will obtain a soft or liquid soap, whereas if it is provided by a sodium hydroxide, it will produce a familiar solid bar soap. In ancient time, soaps were produced by boiling animal fats or vegetable oils into soda or potash. To put it in simple terms, non-hydrolyzable lipids are in general the smallest lipid units used to build bigger structures, which are then hydrolyzable. In our agenda today, the most important lipids, and among them, 2 non-hydrolyzable groups and 3 hydrolyzable groups. There is several types of lipids: fatty acids, glycerides, phospholipids, isoprenoids, and steroids. Let's start with fatty acids. They are composed of a carboxylic acid group linked to a long straight carbon chain with an even number of carbon atoms from 4 to 28. To note, bacteria are exceptions with regard to their odd number of carbons and their branched carbon chain. The carbon chain presents hydrophobic properties as opposed to the carboxylic group, which is hydrophilic, that make fatty acids amphiphilic compounds, both hydrophobic and hydrophilic. Fatty acids are more often represented like this. The disc stands for the carboxylic function, and each corner of the chain corresponds to a carbon atom. However, the longer the chain length, the more hydrophobic the fatty acid will be. This amphiphilic characteristic will lead to the formation of discrete tiny clusters called micelles in aqueous solutions beyond a certain fatty acids concentration called critical micelle concentration, with the hydrophilic carboxylic groups pointing outwards, forming the outer surface of the micelle and the hydrophobic tails inside. It is thanks to this property that cellular membranes are existing to allow the compartmentalization of aqueous media. And this also explains soap properties, trapping the greasy dirt inside the hydrophobic part of the micelle. As the soapy water is rinsed away, the greasy dirt goes along with it. Here you can observe the most common fatty acids in animals and plants. The majority of them have a chain comprising 16 to 18 carbons, like the stearic and palmitic acids. However, as you may have noticed with the empty spaces, this table is not complete, even though all carbon chains are represented from 4 to 24 carbons. Well, this is due to another important characteristic of fatty acids. They could be saturated or unsaturated. All fatty acids represented here are saturated. To be unsaturated, they should comprise at least one carbon-carbon double bond. Thus, instead of the simple bond in the carbon chain, you can find double bond. But what does that really mean? That means that if you consider the complete molecule with all atoms represented, the free rotation around each carbon-carbon bond renders the fatty acid molecule extremely flexible. Due to the steric limitation imposed by the hydrogen atoms around the carbons, the most stable conformation is the linear one. Conversely, if you introduce a double bond, you introduce a rigid curvature of 30 degrees in the carbon chain, which will interfere with their compaction property. Two conformations are then possible, depending on whether or not the remaining 2 hydrogen atoms adjacent to the double bonds stick out on the same side of the chain, cis or trans. The rigidity of the double bond freezes its conformation, and in the case of the cis isomer, causes the chain to bend. whereas the trans conformation does not cause the chain to bend much, and their shape is similar to the straight saturated fatty acid. Fortunately, the cis conformation is most naturally occurring, and it's in fact a good piece of news, because unsaturated fatty acids in cis have a beneficial effect on health, reducing cardiovascular diseases risks as opposed to saturated or unsaturated fatty acids in trans. If we go back to our previous annotation, double bonds are represented like this. You may know that the term omegas that we often find advertised in food, like omega-3, extolling their virtues, is a biochemical nomenclature that permits to localize the first double bond. As omega is the last letter of the Greek alphabet, this implies that we should start to count from the opposite extremity of the carboxylic group. Omega-3 means that the first unsaturation concerns the third carbon from the distal extremity of the fatty acid. If there are other double bonds for polyunsaturated fatty acids, they are located in general every 3 carbons away. The presence of cis bonds can affect the melting temperature of the membrane or of the fat. Indeed, saturated fatty acids can be aligned for their length given their straight linear structure, thus allowing them to be closely packed. Nonetheless, the presence of curved unsaturated fatty acids will create empty spaces without contact, limiting the compaction of lipidic structure and increasing their fluidity. Let's go back to our completed table with both saturated fatty acids, most of the time solid at room temperature, that are found mainly in animal fats and in some vegetable oils like palm oil and coconut oil, and unsaturated fatty acids, mostly liquid at room temperature, like the vast majority of vegetable oils. Especially 2 of them are called essential fatty acids because we can't make our own, whereas they are required by the humans and other animals' metabolism. The alpha-linolenic acid, an omega-3 fatty acid, and the linoleic acid, an omega-6 fatty acid. However, fatty acids are rarely free and are in fact major constituents of more complex structures called glycerides or acylglycerols, or more generally fats. Glycerides are esters formed from glycerol and fatty acids. Thus, glycerol presents 3 hydroxyl functional groups which can be esterified with 1, 2, or 3 fatty acids, also represented like this to simplify. And these esterifications will give rise to monoglycerides, diglycerides, or triglycerides. Vegetable oils and animal fats contain mostly triglycerides but can be broken down into mono and diglycerides and release free fatty acids. It is important to note that the triglycerides' biochemical properties are clearly depending on their fatty acids composition. They are often different within the same triglyceride molecule. Here you have 2 examples of fatty bodies composed of different kinds of fatty acids. Therefore, a preponderance of unsaturated fatty acids will decrease melting temperature, while conversely, too many saturated fatty acids will solidify fatty bodies at room temperature. Thus, butter is composed of triglycerides with around 30% of unsaturated fatty acids, whereas they represent 90% of fatty acids in olive oil. This explains why one is solid and the other one liquid on your table. The 3 fatty acids within a triglyceride can present different chain length and variable numbers of double bonds. This triggers a huge number of possible combinations for one molecular fat. Derivatives of triglycerides, phospholipids, are the source of cellular membranes of living beings. The structure of the phospholipid molecule generally consists of a diglyceride linked to a phosphate group, that is to say, one glycerol molecule, a phosphate group, and 2 long carbon chains of fatty acids. For the anecdote, the first phospholipid has been identified in 1847 by the French biologist Théodore Nicolas Goblet. Interestingly, the phosphate group can be modified with a simple organic molecule, like here in the blue area. There are 4 different groups possible, which will determine different name and functions. In this example, it's a choline. Let's forget about the name of fatty acids. Here we have a phosphatidylcholine, but you can also find phosphatidylserine, phosphatidylethanolamine, or phosphatidylinoxidone. Obviously, all these different phospholipids play specific key membrane functions. Sometimes there is a sphingosine instead of the glycerol and one fatty acid. The molecule is then called sphingophospholipid, which is slightly different of phospholipids and is mainly found in brain and neuronal cells. The peculiar amphipathic composition of phospholipids generates a hydrophilic head containing the phosphate group and the glycerol, and a hydrophobic tail with the two fatty acids. These properties allow phospholipids to play an important role in the phospholipid bilayer. In biological systems, the phospholipids often occur in bilayer, such as cellular membranes. Lipid bilayers come up when hydrophobic tails line up against one another, forming a membrane of hydrophilic heads on both sides, separating two aqueous compartments, the base of cell creation and of their compartmentalization. Let's see the next group, the isoprenoids. Isoprenoids, also called terpenoids, derived from 5-carbon isoprene units. Among them, we notably find phenol, rubber, but also many varieties of fragrant substances and essential oils produced by plants. And of course, the vitamins. Vitamins are vital nutrients that an organism requires in limited amounts. We distinguish water-soluble vitamins that readily dissolve in water, diffuse quickly in living organisms, and are excreted as easily, from liposoluble vitamins, more easily stored and so more difficult to eliminate. Their concentration should not exceed a certain threshold to prevent their toxicity. There are 4 liposoluble vitamins derived from isoprene units: A, D, E, and K. Moreover, some isoprenoids are specifically produced in some animals and plants. For instance, vitamins A, D, E, and K are not synthesized by animal organisms while they are crucial to their metabolism. Another example, rubber, is only produced by the rubber tree. Finally, from isoprene, you can also form steroids, but we will treat them separately. Steroids are polycyclic organic compounds presenting a hydrophobic core composed of 17 carbon atoms bonded in 4 fused rings. Hundreds of steroids exist in the whole kingdom of life. Steroids include, among others, sterols and their derivatives, cholesterol, phytosterol, steroid hormones like the progesterone, cortisol, testosterone or estrogens, bile acids, and secosteroids, from Latin secare, that means to cut, presenting a broken ring, like the vitamin D. Last, cholesterol in particular is also present in animal cellular membrane, where it could reach 50% of total lipids. There is normally no cholesterol in vegetable and bacterial cellular membranes. That's the reason why we said that vegetable oils are without cholesterol, although they have similar molecules. Well, we have described the main lipids making up living beings. Let's know their functions. Like carbohydrates, they are food for cells. Fats, and in particular triglycerides, have high caloric values and usually yield twice as much energy per gram as carbohydrates. It's considered that carbohydrates are used for short-term storage, while fats are used for long-term storage. That's why an average person can deplete his carbohydrate reserves in a working day, but it's much more difficult to exhaust or at least decrease the 16 kilograms on average of fats. In animals, fats are stored in specific cells called adipocytes. Through phospholipids, lipids are also crucial building material of cellular membranes, as well as their compartmentalization. Along the same line, lipid composition within a membrane will affect its properties and functions. Lipids are also remarkable insulating materials. Their presence in subcutaneous tissues and around organs enables a good thermal insulation. Additionally, at the cellular level, they provide both electrical and mechanical insulations. Lastly, lipids play an important function in cellular signalization. Steroids in particular may act as messengers to transmit information from one cell to another. They can anchor some molecules to cellular membranes or be involved in enzymatic reactions. Finally, lipids have key function in vision thanks to the retinal, an isoprenoid. Here we are. Now we do know the most important points about lipids. Thank you for following me, and don't hesitate to comment, like and share this video. See you soon.